Hey, my name is Boddum, and I'm from Sophion Bioscience, a biotech company producing automated PET clamp systems for high-throughput electrophysiology. I'm happy to present a study where we developed electrophysiological assays for large molecule drug discovery. We evaluated snake neurotoxins and the neutralizing potential of recombinant antibodies. It was a collaboration between us at Sophion and the Tropical Pharmacology Lab at the Technical University of Denmark, who did the antibody discovery and development. Currently, more than 90% of approved drugs are small molecules, which are smaller than 1 kilodalton. However, large molecules are rapidly rising in prominence and importance. This group, however, is often expensive and scarce and can be tricky to work with. They degrade fast and can be sticky. Sophion's two automated PET clamp systems, the QPAT and the Cube, allows for 48 and 384 PET clamps recording in parallel. So far, they have been used extensively in small molecule drug discovery. However, the use of APC systems for large molecule characterization has been less common. Therefore, we made this case study where we work with snake toxins and recombinant antibodies to highlight the performance of these two PET clamp systems working with large molecules. Snakebite is a highly neglected tropical disease happening almost 3 million times a year, causing hundreds of thousands of deaths and even more amputations. Currently, the only treatment is animal-derived serum. However, a novel type of antidote is currently under development. It is based on recombinant human antibodies engineered to bind neurotoxins and thereby neutralize their toxic effects. To evaluate neutralizing antibodies, we patched human-derived cell lines endogenously expressing the nicotinic acetylcholine receptors. This receptor is the target of one of the most medically relevant group of snake toxins, alpha neurotoxins. With these setups, we could study the neutralizing effect of different antibodies. First, we made an acetylcholine dose response experiment. And here you see the response to increasing concentrations of acetylcholine. This was done to establish appropriate concentrations required to activate 80% of the receptors. This concentration was used in the following experiments. Next, we confirmed that alpha-copratoxin, a neurotoxin from a cobra, could inhibit the acetylcholine-mediated current. Again, based on the dose-response curve, the IC50 value was found and used for the neutralizing experiments. To determine whether a range of different IgGs could neutralize the inhibiting effect of alpha-copratoxin, 7 different IgGs were tested. Indeed, this inhibiting effect of the toxin was prevented by the IgGs. Furthermore, the IC50s of the IgGs could be determined, which made it possible to rank the IgGs according to their neutralizing effect. However, each snake species has a unique venom profile consisting of several dozen different toxins, and in addition, you often do not see or recognize the snake species that bit you. Hence, antibodies with cross-reactivity is highly desired. To evaluate cross-reactivity, we employed the Cube, which is a system that can run 384 experiments in parallel. In total, 6 different neurotoxins from 4 different snake species were tested. This is the one you see in the top row. And they were tested against 15 different antibodies, which is what you see in the columns. And the play view represents a heat map showing the neutralizing ability of the antibodies and clearly indicate whether an antibody is highly specific to only one single toxin, like this one or this one, or possesses broader neutralization capacity as seen down in this group here. And this demonstrated that cross-reactivity can be achieved. So, in summary, the study resulted in the discovery of a range of antibodies recognizing and neutralizing the effect of toxin from several snake species. This is really an important step in the design of a novel recombinant antivenom. And in addition, it highlights the potential of using high-throughput EFI systems in large molecule research. I'd like to thank you for your attention, And if you have any questions or comment, please do not hesitate to contact me. Again, thank you.